Okay, so here you can see I have my original nucleotide here in black, and I've added more of them. So we had said DNA is double stranded. One strand here, one strand here. Now, what is the strand made up of? It's the phosphate sugar, phosphate sugar, phosphate sugar. Over here, phosphate sugar, phosphate sugar, phosphate sugar. There's two strands. And on the inside here, we had said we have these nitrogen bases. And we had said this might be an A. And if that was an A, that would be a T. And I'll explain why in a minute. And this might be a C. And on this side, it would be a G. So, and so on. So you notice we have two strands here. And if I had a bigger board and I wanted to sit and draw for long periods of time, I could draw this thing really long. But you see we have these little dots between the bases there. And I said this might be an A, and this might be a T, and this might be a C, and this might be a G. Those dots are hydrogen bonds. So if you remember, we were just talking about bonding, right? And we had said polar covalent molecules unequally share electrons. Well, it ends up that adenine, the A, will connect up hydrogen bond with thymine, the T. And cytosine, the C, will hydrogen bond with guanine. So we said DNA, which is going to be storing genetic material, is double-stranded, but you need to be able to make a copy of that. And so we had said, what is hydrogen bonding? It's a very weak interaction between polar covalent molecules. So you may have heard of people talking about DNA unzipping. Well, what we mean is it can pull away, the A's and T's can come apart, so you can make a copy in between there of RNA, so it can leave the nucleus. So we're starting, I hope, to get a feel for this genetic material, which we call DNA. Hopefully you can see that. There it is. And you can see it's a double helix, it's a spiral, and in between there, hopefully you can see the little dots barely, but that are there. And so we have a code that is used to determine your characteristics. Okay, so we have the code. It can't leave the cell's nucleus. And you can make a copy, a single-stranded copy of that code, and it can leave, and that is, we'll talk later on in this section, we'll talk about how that is used to build a protein, actually. Now, so we have this whole genetics side of things that is one of the jobs of nucleic acids. But the other job of nucleic acid is to form short-term energy. And so I'm going to stop the video here, and I'm going to draw an RNA nucleotide on the board and show you a modification of that is the way that within a cell, energy is transferred from one location to another within the cell using a nucleic acid, something called ATP. It's coming up next.